And the story with Alzheimer's starts in the early 20th century. And I think it was in 1908 or 1909 that Alois Alzheimer, who was a neurologist in Munich, described the history of a patient who developed a dementing illness, then Alzheimer actually looked at so he at that time neuropathology, which is the discipline of looking actually at the tissues of people with neurological diseases. At that time, neuropathology was mostly done by neurologists. This is still the case in some countries. In Italy, for example, neuropathology is typically a subdiscipline of neurology, and in Switzerland, it's a part of pathology. And so Alzheimer actually made some histological sections of his patient in the brain, and he essentially described two things. You know what he described, no? What are the two main features of Alzheimer's disease? You must know that. That could actually be an exam question. Yeah, sorry? No. You can't diagnose loss of memory on a tissue section. Yeah. Beta amyloid is cool, it's very nice. It's not what Alzheimer's saw, because there was no, this was in 1908. Nobody knew what was A beta. So he had two features. One is called the neuritic plaques, and the other are called tangles. So the neuritic plaques is essentially, now we know what it is, but this is really what Alzheimer saw. He saw that in the brain of people with Alzheimer's disease, he saw there were plaques of, somewhere between 50 and 200 microns in diameter, even larger, and this was, you have to realize that in the early 20th century, Munich was the absolute capital of the medical neurosciences. I mean, the stuff that people were doing in those days were amazing. It was like Silicon Valley for artificial intelligence, and in fact, everybody who was doing interesting stuff in the neurosciences was actually working in Munich. And Nissl, the guy who invented the Nissl stain with which you can stain neurons, was there, and Bielszowski, who invented a way of staining neuronal processes with metallic gold, and they were all there. And Alzheimer was actually the director of the institute, and it was a very good morphologist. So he found actually these plaques. And now I have to say that the amazing thing is that 120 years later, the neuritic plaques are still the golden standard. It's how you diagnose Alzheimer's disease nowadays on morphology. And yeah, of course, you can diagnose dementia clinically, but the problem with that is that if you only go by the clinic, the most common cause of progressive dementia in the elderly is Alzheimer's disease, but there are many other causes, and let's go through them. What are other causes of slowly progressive or rapidly progressive dementing illness in humans? What comes to your mind? It's very early in the day, I know, but you have to wake up. Yeah. Maybe Parkinson. Okay, so not completely wrong, but almost completely wrong. And so why is that not a perfect answer? What are the diagnostic criteria and the salient signs and symptoms of Parkinson's disease? Yeah. That's wonderful. Substantia nigra is a great hint. I was talking about signs and symptoms, and signs and symptoms again is the opposite of what we were talking before. It's something that you cannot diagnose on a tissue section. You have to actually look at the patient. And what do patients with the Parkinson's disease have? Yeah. Tremor, yes, tremor is correct. Other things? Yeah, very good. Recur, akinesis, that's absolutely excellent. That's the trials of Parkinson's disease. Dementia, is that an issue? No. So let's agree that Parkinson's is not typically cause of progressive dementia, but it's actually more complex than that. And some people with Parkinsonoid syndrome can develop dementia, and the pathogenesis is actually very similar. And I just want to mention that this goes now again a bit into the molecular things, and but what is the protein that typically accumulates in the brain of people with Parkinson's disease? No, it's not tau. It's a protein called synuclein, alpha-synuclein, and it turns out, and that is kind of a bit, I mean, we'll talk about this in a minute, but the fact with synuclein is that synuclein, when it accumulates in the substantia nigra, it will induce a disease called Parkinson's. So it will kill actually the neurons of the substantia nigra. And but synuclein cannot only accumulate in substantia nigra. There are cases in which it accumulates pretty much everywhere, including in the cortical neurons. And then it causes something else. Actually, in the substantia nigra, synuclein causes aggregates, protein aggregates, deposits. They're much smaller than the plaques. They are inside the neurons. The plaques are outside. And so they're not neuritic plaques. They're intracellular aggregates, and they are called some bodies. What kind of bodies are in the substantia nigra? Yes, the Lewy body. You're good. I like that. So yes, these are the Lewy bodies, and now the thing with the Lewy bodies is that when they are restricted to say, and well, I mean, it's even more complex than that. They are not really restricted to the substantia nigra. They never are. You can find them in the locus ceruleus and in other places, but when they are restricted more or less to the diencephalon, mesencephalon, then you get a syndrome of Parkinson's, and there are very clear neurophysiological reasons for that. But there are so-called synucleinopathies, which are diseases in which synuclein accumulates, but it not only accumulates in the substantia nigra, it accumulates also in the neurons of the cortex. And then if the neurons of the cortex are damaged, then you get a dementing illness. And this is called dementia with Lewy bodies, DLB. And so the synucleinopathies are essentially, there are three syndromes. One is Parkinson's disease. The other is another disease called multiple systems atrophy, which is a terrible disease, very rapidly progressive. It's always lethal. It's actually the worst of the synucleinopathies. Multiple system atrophy is essentially a disease of the brainstem, and then there is dementia with Lewy bodies, which is generalized. When you get Lewy bodies in the entire brain, and that of course gives rise to dementia. So and for that reason, I was saying that saying Parkinson's is the cause of dementia is not entirely correct, but it's also not entirely wrong because there is a continuum. There are patients who start with a Parkinson syndrome, with a tremor and the rigidity and everything you know about Parkinson's, but these patients progress to a dementing illness. Other patients have only the motor syndrome. They have trouble in starting movements, so that is actually very typical. But and the tragic thing is that these people, they are perceived by their environment as maybe as demented because they also don't have facial expressions. They can't smile or laugh and so. And of course, this leads to the perception of psychological problem. But in reality, they are cognitively perfectly fine, and the problem is a motor problem. But that's not always the case. There are also patients who develop dementia. So things with synuclein and Parkinson's are complex. Let's go back to the differential diagnosis of dementing illnesses. What else? Yeah, go ahead. Right, so you need to be more attentive. The question is, what are the causes, the typical causes of progressive dementing illnesses in humans? And we have started with Alzheimer's, and then I have explained that Alzheimer's is one possible cause, potentially also the most frequent one, but there are other causes as well, and we were going through these other causes. And this is very important also clinically, because if you have a patient in front of you who is cognitively impaired, I mean, the most relevant question that you have to ask yourself is this a treatable entity or not? Because if it is not treatable, then yes, it is academically important to find out what is the diagnosis. Not only academically, I mean, it's also, because there are hereditary syndromes, so you may have to do family counseling, human genetics, and so. But of course, the most important question is it something that I can treat? And you have to recognize that, because if you don't, then then this is malpractice. So you really need to know which causes of dementia can be treated and which can't. So let's go through them. Yes, from FTLD, frontotemporal lobe dementia is also as the name already says, it's a very rare event. There is a different protein involved. The protein involved there is called, anybody knows? It's a strange name. It's called TDP43, and TDP means TAR domain binding protein, when the TAR is actually a part of lentivirus HIV, and it's acting regulatory sequence of HIV, and this is how TDP was discovered in the first place, and then it was forgotten because it was not so important, and then actually the story is actually fascinating. And then people started, there is another neurodegenerative disease which is non-dementing, but it's a disease of the motor neuron in the spinal cord. And these people end up with, actually, not only the spinal cord, it is actually it's a two neuron disease. The motor neurons in the ventral horn of the spinal cord degenerate, but also the primary motor neurons in the precentral gyrus of the cortex, which are essentially the first synapses of the first neuron of the motor system. They also degenerate. Which disease is this? ALS, amyotrophic lateral sclerosis. Very good. So ALS is not a dementing illness. It's purely motor syndrome. It's a terrible syndrome. It's a horrible disease, and people eventually suffocate. They cannot breathe anymore. Then they have to go onto respirator. It's horrible. And in fact, I already have two friends who are actually dying of this, and there is nothing to be done. And I tell you, it breaks my heart. And and it's not such an uncommon disease. It's not very common, but it's also not very rare. And so that is ALS, amyotrophic lateral sclerosis. And then people notice that in ALS, there are also small aggregates in the neurons. So if you look inside the cells of the neurons, there are little aggregates that appear there. And the question is, what was it? And this is very, very hard thing to do. And even nowadays, with nowadays technology, it's very hard. And then there was a scientist her name is Manuela Neumann, and she's a neuropathologist, and she was a postdoc in the laboratory of John Trojanowski and Virginia Lee. This is a couple. They have discovered almost all of the proteins involved in neurodegeneration. It's just unbelievable. And when Manuela went to the lab of John and Virginia, she was given the project of isolating the protein that made up these little aggregates in the motor neuron of patients with ALS. And that was an enormous task. And what she did essentially was to, you can fractionate. You can take the spinal cord of patients with ALS, then you can centrifuge the junk off, then you can do gradient centrifugation where you use a gradient, a density gradient where aggregates of different sizes and of different densities will segregate differently at various levels in a gradient where you have very high density at the bottom and low density at the top, and this way you can enrich. But the problem is that these aggregates were very, very, very little. It's just in trace amounts. I mean, you have a whole spinal cord and you have milligrams or micrograms of aggregates. It's very difficult to purify. So so eventually, what eventually Manuela did was to have a partially purified fraction, and then she made monoclonal antibodies. That is, she took the enriched fraction and she injected into mice, and she recovered, and then the mice developed an immunity against whatever was injected. But whatever was injected was, of course, was a mixture. I mean, there were the ALS aggregates, but there was a lot of stuff, other stuff as well. And but once you have the antibody, you can make monoclonal antibodies out of them. So you can take a single B cell that will make a single antibody of a single specificity, and then you can use this to stain the sections of the spinal cord with the people, and you can ask whether that antibody will actually recognize the aggregates. Huge amount of work. It worked. It worked, and she found an antibody that was actually recognizing the aggregates, and then you can use this for immunoprecipitation. That is, you can bind the antibody to a solid phase, like, for example, beads, magnetic beads or agarose beads, and you can use it to actually capture the aggregates and pull it down. And then, and now you can wash it very, very harshly because everything else will be washed away. And now the only thing that remains attached to the antibody is your protein of interest. And now you can actually ask, what is this protein? And what is a method that you can use to determine the identity of a protein? I mean, a protein consists of, it's a polypeptide. It consists of various amino acids. How do you determine the sequence of and the composition of a protein? No. PCR is for nucleic acids. This is, no, not PCR. Yeah. Sorry? SDS page. So what is SDS page? What does it mean, actually? What does the abbreviation SDS stand for? Okay, SDS means sodium dodecyl sulfate. It's PAGE means polyacrylamide gel electrophoresis. So it's a way of separating proteins. So in that you are right. You can separate and you can find so let's say you have an unknown protein. You can run it on a polyacrylamide gel and the protein will migrate on the gel, and large proteins will migrate less quickly than small proteins, and this way you can actually determine the molecular weight of a protein. You won't be able to determine the sequence, and you only can find out what is the molecular weight. You cannot determine the sequence from that. So how do you do it? How do you determine the sequence? Yeah. Sorry? Maldi, very good, very good. Now we are coming closer to the truth. So what is Maldi? Mass spectrometry. So what does the acronym Maldi stands for? Matrix assisted laser desorption. So Maldi, Maldi is one technology, but essentially there are a number of technologies that are, the umbrella term is mass spectrometry. So what is mass spectrometry? You're good. You're coming to the right thing. You have the right intuition. So what do you do with mass spectrometry? That's why it's called MALDI-TOF. And what does TOF mean? Time of flight. Very good. So that is with MALDI-TOF, what you do is you have a big protein, and then actually you do several things. You actually fragment it into smaller fragments, and then these fragments are run into a vacuum tube, and what you do is you measure the time. A bigger fragment will fall off the tube pretty rapidly. A smaller fragment will fly all the way through, and this way you can measure the exact molecular weight of each fragment, and then it becomes, so in the end, you also end up with molecular weights, nothing else. So it's the same thing as PAGE, but the molecular weights are super precise. You can easily discriminate isotopes with a difference of one Dalton with MALDI-TOF. So it's very, very precise. And now you can imagine that if you have a protein and you do random, let's say the protein has 100 amino acids, and you do now break it down randomly into anything from 1 to 10 or 20 amino acids, and then each fragment has a specific molecular weight. Now you still can't actually directly infer what is the sequence of the protein, but because you have so many different fragments, you can start calculating. Okay, what does it fit? No, and then you can reconstruct the puzzle, and eventually you get the entire sequence. So that's more or less how mass spectrometry works. And of course, nowadays every protein of the human genome is sequenced, and therefore you don't actually even need the full sequence from mass spectrometry. You just need small fragments, and these, if you can sequence already a fragment, this will already be enough to interrogate the database and to find out what is the match, what is the protein that is matched. So that is how this whole thing works, and that is what was done. And then Manuela found that the protein recognized by her antibody was TDP43. And then she became very famous, and her paper was cited many, many thousands of times. It's one of the most cited papers in the neurosciences. And then she wanted to come back to Europe because she's German, and then I offered her a job as an assistant professor in my institute, and she came. And so So she was actually my first assistant professor, but very, very successful. And but she didn't stay long because she was so famous, everybody wanted to have her, and so she was recruited to the University of Tübingen, where she's now the chair of the Department of Neuropathology, and very successful scientist. So that is the one story. Now, why did I tell you all this thing about ALS? ALS is not a dementing illness, but today we are going to talk about dementia. Now, the thing is, there is another disease which is called frontotemporal dementia, and now FTD, and it turns out then in FTD you have the same thing. You have these little aggregates, but now they are not in the spinal cord. They are not in the precentral gyros. They are everywhere. And most affected is the frontal lobe, and this is actually what causes frontotemporal dementia. It's so it's again it's a FTD. It's an TDP43 related pathology, and again there is a continuum. There is, on the one hand, you have pure dementing illnesses. On the other hand, you have pure motor neuron disease with a paralysis, and then you have anything in between. And so that is, and that I think is something very important that you can that you see time and over in neurodegeneration research, that you find you start with a very rare disease, and then you find a mutation that is causing this disease, and then you ask, is this mutation common in other neurodegenerative diseases? And then you find actually that it's very common, and that it plays a role in the sporadic forms, and this is essentially what happened here. And something very similar happened also with synuclein, because with synuclein the story is also fascinating. The existence of the Lewy bodies was known for a long, long time, ever since people started looking at the microscope as the substantia nigra, and the substantia nigra degeneration is, the neurons of the substantia nigra, they project the axons of the neurons of the substantia nigra. They project, where do they go? Where do they go? This is neuroanatomy. You must have learned this in your second year of study. Where do the projection of the substantia nigra go? Yeah. To the striatum. Very good, very good. So these are if the neurons of the substantia nigra disappear, then you have an imbalance in the neurophysiology of the striatum, and this is actually what causes the Parkinson's disease. And for whatever cause, you can. There are many ways to kill the neurons of the substantia nigra, and one way is to fill them up with Lewy bodies, and then you will destroy them. And in fact, the substantia nigra becomes what I would call substantia alba. The substantia nigra means in Latin the black matter, but it becomes white. You don't, in a patient with Parkinson's disease, you don't see it anymore. It's gone. And but it's not the only way to kill these neurons are dopaminergic, and in fact there are poisons that will kill specifically the dopaminergic neurons in the substantia nigra, and they create a disease that is identical to Parkinson's disease, but it has nothing to do with synuclein. And in fact, there was a famous disease where I think it was in California that they were making designer drugs. I mean, that's what people do in California, and they were making designer drugs, and one drug was contaminated with a chemical called MTPT, NT, and I don't remember what the acronym stands for. And but this is essentially a contaminant that can be taken up by the dopamine transporter in the neurons of the substantia nigra, and it will be metabolized in a terribly toxic reactive species, and this kills the neurons of the substantia nigra immediately. So these poor people who actually took this drug, on the next day, they had a full-blown Parkinson's disease. These were but they were in their 20s, and there are other so terrible story and terrible disease, and identical to Parkinson, but nothing to do with synuclein. So this shows you essentially, and also in mice and in rats, you can actually cut the nigrostriatal fibrils, the axons that goes from the substantia nigra to the striatum. You can actually cut them, and then you get a full Parkinson. You can even get a hemi-Parkinson. You can cut them on one side, and then you have a disease that where the Parkinson's disease is limited to one side of the body. So, so this is the neurophysiology of it. But now let's go through the biochemistry and the genetics of it. Now, so synuclein. So, how did we end up finding out what is what is synuclein and why is it causing the formation of Lewy bodies? Now, the thing is, I think that the first person who actually identified synuclein was Michel Goedert, who is a scientist originally from Luxembourg, but he works in Cambridge since many decades. And now he had a paper in Nature in the mid-90s, where short paper where he described, and again, I'm pretty sure it was an antibody, so very similar to the TDP story, where he described an antibody that would recognize recognize the Lewy bodies. But then actually, what I think was one of the major breakthrough was a human geneticist called Polymeropoulos, and he found a family in northern Greece, and this family had hereditary dominant, autosomal dominant Parkinson's disease. Everybody who had in a family had Parkinson's, 50% of the progeny had Parkinson's, so it was very clearly familial, and it was inherited as a dominant autosomal trait. And then he actually identified the gene, and the gene was a duplication in the gene encoding for synuclein. So in these people, there was on the synuclein locus, the synuclein gene was actually duplicated. So these people actually had 3 copies. Each one of us had 2 copies of synuclein. These people had 3. And having 3 copies of synuclein sufficed to provoke the disease actually in very young people in this family. So, and that is very strong. I mean, if you have genetic inheritance and you have 100% penetrance, and everybody who has the triplication, or actually the duplication, develop Parkinson's, and everybody who doesn't have it in the family does not develop Parkinson's, then you have causality. Then it's almost impossible that it's something else. So, and then actually, but this was one family in the entire world. It was, I mean, now in the meantime, we know also there are a couple of other families. They probably all come from the same founder. I mean, there are some people in southern Italy, but you know, they probably, you can trace them to the same to the same founder effect. And so this is super rare. Yeah, but you know what? If you make an antibody against synuclein and you take the substantia nigra of anybody with Parkinson who died of Parkinson here in Zurich or whatever, and you stain it with the synuclein antibody, it's full of it. The Lewy bodies in the sporadic form of Parkinson's disease consist of synuclein. So the synuclein was discovered in an extremely rare form of hereditary autosomal dominant Parkinson's disease. But it turns out that the synuclein aggregates are the cause of sporadic Parkinson, which is the second most common cause of neurodegeneration. So it's So, and again, this is the point that I'm trying to make, that the study of rare variants of human genes, these are rare things, but then they can teach you stuff that is important for the entire population. And so it's worthwhile. I'm saying this pointedly because I'm reading, people, I mean, pharma guys, and so, yeah, it's not commercially interesting to study rare disease. Let me tell you this complete bullshit. I mean, you can make fundamental discoveries about very common diseases by studying rare diseases. So it's really something. So one example is Parkinson, but now let's go back to Alzheimer's. Unless you want to ask me something about TDP and the synuclein. I know, before we go to Alzheimer, I want to share another story about Parkinson's because it's also super interesting. So, now, there is an enzyme that is called glucocerebrosidase. Ever heard of it and what it does? So glucocerebrosidase is a hydrolase that takes a glycolipid called glucocerebroside and cleaves it into its moieties, a sugar and a lipid. And so that's why it's called glucocerebrosidase. And it's a lysosomal enzyme. It resides in lysosomes. Now, it's an important enzyme. And if you have a homozygous mutation that inactivates glucocerebrosidase, you end up with a lysosomal storage disease. It's a very severe syndrome that appears in infants, is progressive, and is lethal. What is the name of the syndrome? I should say that I'm not a fan of naming syndromes according to people because, then all kinds of bad things happen. Then then you honor the discoverer. Then it turns out that the discoverer was actually a murderer or something like that. Then you regret that you actually have named the syndrome after the. So, but I don't know. I mean, it's called Gaucher's disease, and Gaucher's, and so the gene is called GBA. It's glucocerebrosidase, and the homozygous mutation, so the null allele, people who have a null allele, they develop a thing that is called Gaucher's disease, and Gaucher's disease is a lysosomal storage disease. Now, what turns out to be the case is that if somebody has Gaucher's disease, this means that his parents are heterozygous, no? Because it's an autosomal recessive disease. So you need both alleles to be knocked out in order to have the disease. But you can only you can have a spontaneous mutation, but the more likely thing to happen for you to have the disease is that both of your parents are plus minus. They are heterozygous. We agree on this? You have a question? Right. So it turns out that the parents have a much higher risk for Parkinson's disease than the general population. And now this is interesting. So why is it? The parents, they don't have a storage disease because, if you have only one copy of the enzyme, that's enough for the catabolism of glucocerebrosidase. And so people don't get the storage disease. But, then they actually at pretty old age, but, up to 30% of them develop Parkinson. So there are two questions here. One is why do they develop Parkinson? And then if only 30% of them develop Parkinson, why? What happens to the other 70%? I mean, so why is it so variable? And so this is super interesting. And And however, I can't tell you the solution because we don't know. I mean, it's actually something that I'm doing in my lab. I think it's super interesting. And so for me, I mean, I just tell you a couple of things, how I think one could approach this, and which actually we are doing. One question is, okay, I mean, if only 30% of the people develop Parkinson, this means that GBA is not the whole story. I mean, there has to be more, and so there has to be modifiers. So maybe there is a second gene, and then it's a glucocerebrosidase plus this second gene, and then you have, if you have both, you get Parkinson. If you have only one, you don't. So that may be, for example, so there may be a complex genetics behind it. So what is the second gene? So I thought that one way of looking for it would be to do a genome-wide screen. And one idea is, okay, we take cells and we inactivate one by one every single gene of the genome. And then we ask, what happens to GBA? Does it go up? Does it go down? I mean, one possibility is that there are factors that will influence. So maybe you are heterozygous for GBA, so your body makes 50% of the amount of the enzyme that wild-type people would do, and that may not cause Parkinson's. But then if you have something, if there is something that is required for GBA expression, and that is missing, then you go up to 25%. But a person who has two alleles would stay at 75%. So that would be so one question is, what is controlling GBA? So you can so the idea is, okay, let's take every single gene of the genome, inactivate it one by one, and ask, do we find anything that controls the expression of GBA? And then if you find it, does it play a role in Parkinson's? So how many genes does the human genome have? 20. Wow, that's pretty good. Yes, yes. I mean, actually, yes, it's around, I mean, if we talk about the protein coding genome, yes, it's around 20,000. If you then take the so-called non-coding, the long non-coding RNAs and the microRNAs, and so then it's actually many more, but the protein coding genome is I mean, the protein coding genome that can be addressed with relatively simple methods is around 19,800, something like that. And so how do you inactivate? So the idea, I mean, if you want to find a gene that is controlling GBA, then probably the best thing is you just inactivate each one of them step by step, and you ask which gene, if inactivated, allows or leads to a drop in GBA activity. And GBA activity is relatively simple to measure because if you just grow cells, you can give a fluorescent substrate. It's something that looks very similar to glucocerebroside, but when it's cleaved, I mean, it contains actually a fluorochrome, a substance that will actually fluoresce, but it does not fluoresce because it's quenched when it is conjugated to the glycolipid. Now, when GBA cleaves it, then it becomes unconjugated, and then it fluoresces. And then you just measure the fluorescence using a spectrophotometer. So it's a simple fluorimetric assay for enzyme turnover. So that part is relatively easy. But how do you inactivate serially 19,800 genes? What is the method that you would use for that? Yeah, CRISPR, great, great. Yeah, you know everything. So why aren't you working in my lab? I mean, well, no, it's CRISPR. Yeah. So that is one possibility. So CRISPR, you can do many things with CRISPR. I mean, does everybody know what is CRISPR? Does anybody not know what is CRISPR? You know about it? Everybody does? You're sure? I mean, you can, there is no shame. I mean, you can ask me. Okay, right. So there are many ways of using CRISPR for experimental biology. What CRISPR does is essentially there is an enzyme a nuclease. It's called Cas9, and it's a programmable nuclease. So essentially, Cas9 will you can couple it to short sequences of RNA called single guide RNAs, sgRNAs, and the single guide RNA will bind to Cas9 and will bring it to a specific part in the genome that is complementary to the sequence of the single guide RNA. So this is why. So this is how you can program the Cas9 to go to a specific locus in the genome, and then it's an endonuclease. It just cuts. It makes. It cuts the DNA at that place, and then repair enzymes come in and they actually repair the cut, but they do this inefficiently and they make mistakes, and therefore, in many cases, you end up with disruption of the gene, and this is how essentially Cas9 works. So it's not perfect. There is a certain randomness of it. It's also not 100% efficiency, but it works. So that is how you use the Cas9 nuclease. Another possibility, which I think is even more interesting, is now the complex of Cas9 with a guide RNA essentially is very generic. I mean, you can, if you use enzymatically active Cas9, you can cut the DNA, but actually you can kill the enzyme by making a mutation in the active site. Now you have a Cas9 which is called dead Cas9, so it's or dCas9. Now dCas9 is the same as Cas9, but it does not cut anything. And but it still goes if you put the correct guide RNA. It will go wherever you want it to go in the nucleus and in the genome. And now you can do some cool stuff. No, I mean, you can, for example, fuse the dead Cas9 to whatever. I mean, you can fuse it to a transcription factor. What is a transcription factor? What is a transcription factor? All right. What is a transcription factor? Anybody wants to bring in? Yes. Exactly. It's actually what the name already says. The transcription factor essentially is, these are proteins that regulate the transcription of genes. So what they do is they actually bring in the RNA polymerase and essentially enhance or promote the transcription of the gene of interest. And the transcription factor typically recognizes sequence in the promoter of the gene, in the upstream section of the gene, or 5' section, and then transcription factor come in, recruits the RNA polymerase, transcription starts. No? And so let's say you want to activate whatever gene you might be interested in. Well, you can use CRISPR to do that, and it works actually unbelievably well. What you do is you take your dead Cas9, so you now don't want the Cas9 to kill the enzyme, to kill the DNA, or to kill your gene of interest. So you take the dead Cas9 that has no intrinsic enzymatic activity, but you fuse it to a transcription factor. Can even fuse it to 3 transcription factors to make things stronger. You can take a dead Cas9, put 1 transcription factor, then when it's finished, you put the next one, and then another one. So you have a chain of 3 transcription factors. You put in the guide RNA, it goes straight to your gene of interest, boom, you get a 1,000-fold activation of expression of that gene. And you can do this for any gene in the genome. So that is a pretty powerful way of using CRISPR to modulate the transcriptional machinery. You can do other things as well. You know what is a promoter, no? You know what is the promoter of a gene? You do? Not really. So why don't you explain what is a promoter? Yeah, I mean, actually, transcription starts actually downstream of the promoter. So the promoter is actually everything that all the regulatory elements that are upstream. And yeah, it can be 500 base pairs, can be 1,000 base pairs. It's not well defined how long it is. There are also enhancers, transcriptional enhancers that can be very far away. They can be even a megabase away. And but the promoter is actually the place where if you don't have the promoter, the gene will never be transcribed. So the promoter is required actually to get the transcription going. And now, yes, if you don't have, so transcription factors bind to specific sequences in promoter. No? So there are target sequences which attract the transcription factors, and but promoters can be actually switched off epigenetically. So there are ways to actually shut down the transcription of a gene without changing the actual genomic sequence. And what is, how do you switch off a gene epigenetically? Sorry? I can't hear you. Yes, methylation, absolutely correct. So you can, if you methylate a gene, then you will switch off. Now, tell me a way of you doing this with CRISPR, using the information that I just gave you 10 minutes ago. Yeah, there is. I mean, that is something else. I mean, there is a methodology called antisense oligonucleotides where you can actually make, that is actually so-called locked oligonucleotides. So these are chemically modified DNA, not RNA, DNA nucleotides that will bind to a complementary, it will be complementary to the nascent RNA, and that will hybridize with that RNA. And this induces actually another system that will eventually degrade the RNA, but that's not related to CRISPR. It works. It can work actually very well, and but it's a different thing. But you were right in saying that methylation can switch off, that you can actually switch off a gene by methylating the promoter very effectively. It will, once the promoter is methylated, the transcription factor will not bind to it and the transcription is gone. It's a very effective way of switching off gene. Now, tell me how to do it with CRISPR. Yeah. Absolutely. And what is the enzyme? Very, yeah. Sorry. Yes, yes, yes. Yes, S-adenosyl methionine is not an enzyme, it's actually a metabolite, but yes, it's a methylation group donor. And the enzyme is called DNA methyltransferase. And essentially, that's what it does. It takes the methyl group and it attaches it to the nucleotides in the DNA. And the result is that it suppresses, I mean, this is epigenetic regulation of gene transcription. So the methylation of the promoter will switch off the gene. So now it's, you can do it with CRISPR. You can take that Cas9 and instead of attaching it to a transcription factor, you attach it to a DNA methyltransferase. And now you have a programmable system that will allow you to switch off whatever gene you're interested in. And in the case of GBA, you can do this very systematically, one gene after the other, until you find the genes that control the GBA expression. So that is, these are the two things that we have been doing in my lab. And yeah, we found a bunch of genes that control GBA transcription, and the most interesting thing is that some of these genes are druggable, so you can actually take a compound and activate this pathway, and thereby enhancing the expression of GBA. And this obviously is something that one could hope at some point will give rise to a therapy of Parkinson's. So very exciting. However, let's not get carried away. I mean, from basic discovery, even if you find a compound that works as you would hope, from there to get into something that you can buy in the pharmacy and cure your Parkinson's is a long way. It's a long way, because you know, it's not only about finding the mechanism. Then you have to find actually a compound that is safe, that is tolerable, that it doesn't have too terrible side effects, and the fact that something works in cells does not mean that it works in humans. Even if it works in cells, it has to get to the brain, and many compounds just don't get through the blood-brain barrier. So, from a basic discovery to a clinical application is a very long way. It can take 20 years easily, but you know, if you don't have the discovery, then nothing will happen downstream of it. So the discovery is the first place. Now, for me, they also, I mean, this is more kind of a general comment, but who does what in this chain of things that lead to the production eventually of a drug? I mean, in my vision, the basic discoveries related to pathophysiology, which is what I'm explaining today, they are, I mean, they are best done in academia. I don't see any kind of industrial environment having the motivation and also the endurance to do these kind of things, because these discoveries, they take a long time, and there is not an immediate commercial reward. It's all very idealistic, so industry will never do it. But I think that academia as a whole is very much capable of doing this kind of thing. So this is the part that I think academic labs can and should do. And then once you have established what is your target, when I am a target is kind of pharma speak, it means, okay, I mean, GBA is a target, or synuclein is a target for Parkinson's. It's a molecule, it's an endogenous protein made by your body that can be modulated and will hopefully do something to protect you from Parkinson's. Now, so that's what I call a target. Now, and then you have to develop compounds that will actually do something to this target. And as I said, I mean, discovering the target, I think, is a very academic task. It's I mean, there are examples in which some targets have been discovered by industry, but they're very rare. Most of the stuff comes from university research. And then, however, the next step is really to develop a compound, and that we cannot do, or we cannot do very well. I mean, people in academia try to do that. It almost always fails. And for many reasons, but yeah, pharma is actually has the tools. I mean, for example, I mean, if you go to Novartis or to Roche, I mean, you know, they have libraries, compound libraries with like 2 million compounds in there. So they synthesize 2 million compounds, and they have them all stored in computerized robotic systems. So once you have let's say you have an enzyme, you know, and then you want to find something that will inhibit that enzyme, or in the case of GBA, that will enhance the activity of that enzyme. They have all the robotics to actually test 2 million compounds in a couple of months, do a big campaign, and then randomly. I mean, there is no, you know, there is no, this is really, these are screens. There is no rationale to testing one compound over the other. They just test everything, 2 million things, until they find something that works. And then once they have that, And that is already something that we cannot do because we don't have these libraries. I mean, they are way too expensive, and it's also not, it's boring work, honestly. Stop working, yeah. And so the so I think it is okay that industry does it, and even then we find a tool compound from your 2 million compounds that works. It's still not a drug. Then you have to do what is called medicinal chemistry. That is, you have to take the compound and start modifying it because typically what you found from the library, yeah, it works, but it doesn't work very well. So you need to enhance the potency. You need to make it water soluble. You may need to make a version that will go through the blood-brain barrier, and so on. This is really the so-called medicinal chemistry. That takes a lot of people and a lot of effort to modify, a little, put an oxygen there, a fluoride there, and a chlor atom there, and so on, and try to find it until you get something that works better. And again, it's not really. It's engineering. It's chemical engineering. It's not discovery work, and for this reason, it's also something that, in my opinion, a doctoral student or a postdoc should not do because you should be doing things that are a bit more intellectually rewarding. And but I have to say, it also, I mean, the medicinal chemistry is tough. It's very, very difficult. I mean, you really need excellent people to do that. It's becoming easier because of artificial intelligence. I mean, now you can model things. You can predict structures of proteins with AlphaFold, which got the Nobel Prize a couple of weeks ago. You can predict how a small molecule, like how a drug will interact with its target. And then once you have the atomic details from the artificial intelligence, also from physical data, then you can obviously do what rational drug design. So instead of just taking a random 2 million compounds and finding what works, you can actually design something that is predicted to work on the basis of the structural interactions. And so that is how, and you do this iteratively, and then you end up with what is called compound attrition. No? You start with 2 million compounds, and it's like a funnel. No? Then you screen them, you end up with 100 compounds that have some activity. Then you start modifying them. Then you end up with 10 compounds, and then eventually you have just one lead candidate, what's it called, the lead candidate, and that is the compound that you want to start going into humans. And then you go for phase 1, which means that is only about safety and making sure that you're not killing anybody by giving the compound. And then once you have done phase 1 and you are sure that nothing horrible happens, then you go on to phase 2, where you perhaps treat 100 patients. And now you start looking, okay, do I get a clinical effect? And phase 2 still doesn't tell you the whole story. It makes, because in the end, you will only find out if something really works if you go into 1,000 patients, and then you have strong statistics, and that is phase 3. And that costs $1 billion easily. And so again, it's completely out of the reach of what an academic lab can do. And even then, even after phase 3, you can then all of a sudden it turns out that some people develop strong side effects and so on. This is what's called post-marketing analysis, and that can happen. That then all of a sudden it turns out that something that had all the credentials to become a successful drug eventually had to be withdrawn because of some unpredicted things that happen, and so that's essentially the whole chain of events that goes from a discovery to a drug. And I'm explaining this to you because because one has to be realistic. As a single person, you cannot do the whole thing. You can just do a part of it. So what we do as academia is really the first step, and everything else has to be done by other people with also other financial resources. Right. So. Is this all known to you, or is this something new? You know all of this. Yeah, that's good. But you should tell me, because then I can go faster. I mean, I don't have to tell you these whole stories. Right. So let's go back to Alzheimer's actually. So there the story where I left it was with the neuritic plaques, and I mean, as I told you, I mean, that remains the discovery by Alois Alzheimer's after 120 years remains the cornerstone of tissue diagnostic of Alzheimer's disease. It's if it's Alzheimer, you have to find the plaques. If it doesn't have plaques, it's not Alzheimer's. And so the plaques are really, I mean, it's amazing that something that was discovered with rudimentary techniques so many years ago is still actually 100% valid and can be said of very few scientific discoveries. And so what are these plaques? Well, everybody wanted to know this, but we are talking beginning of the 20th century, so there was no mass spectrometry. So I mean, so what happened was, I mean, back then neuropathology was fundamentally German thing. I mean, most of the scientists were actually in Germany. There was some great neuroscientists in Spain and in Italy, and the two most famous, I guess you will know, the Spanish guy who made these beautiful drawings of Purkinje cells and of microglia. Who was that? Yes, exactly, Ramon y Cajal, and the guy in Italy who was a big enemy of Cajal. Yes, Camillo Golgi. He was the rector of the University in Pavia, which is the city I come from, and they had a big fight, and in fact, they didn't talk to each other. They got the Nobel Prize together, and they didn't talk to each other during the ceremony. They were really totally, and but perhaps because Golgi is kind of because he came from the same town as I come from, and so I should probably be a fan of Golgi. But the fact is, he was pretty obnoxious, and also he was wrong. He was completely wrong, and I mean, Golgi invented the Golgi stain of neurons, which was revolutionary. I mean, I mean, even now, I mean, it's the real beauty of the Golgi stain is that it doesn't stain all the neurons. It stains some neurons sparse stochastically, and this is now really interesting because now there are technologies that allow you to make brain completely transparent. And if you couple this to the Golgi stain, you can actually see the neurons in 3D. And if you stain all the neurons, you won't see anything because there is just, they're too dense. But if you only see stain them sparsely, then they come up beautifully. So it's a great technology. And for that, he deserved the Nobel Prize. But, Golgi thought that there are no neurons, that everything is interconnected with everything else in the brain, and then Cajal actually showed that no, that's not true. That there are distinct cells and they attach to each other with synapses, but they are not just one single plasmodium, and that was the beginning of big fight and big egos and so, and it never healed, but in the end, I have to say that Cajal was right, so using these techniques, I mean, the way Alzheimer's found the plaques was very similar to Golgi stain, only he used silver. It was a silver impregnation method where he actually used silver nitrate, and it's a bit similar to the way how you used to develop old black and white films, before digital, that you had a gelatin with silver nitrate, and then this would, the light would generate metallic silver that is black, and this is how films were made, and this is a similar technology to stain plaques, but then at that time, obviously, it was not clear what these plaques are. The only thing that was clear is that if somebody had dementia, they typically had these plaques. And then for many, many decades, nothing happens. And I think that, I mean, my interpretation is that neuropathology suffered terribly because during the Nazi period. And during the Nazi period, I mean, horrible crimes were committed by medical doctors and pathologists, particularly, and at the institute in Munich, where Alzheimer used to work, the head of the institute was a guy called Hallervorden, who was a monster, honestly. I mean, I have to say, I mean, I was a couple of years ago, I was invited. Now it's called the Max Planck Institute for neuroscience, and I was invited to give a talk there, and they had a kind of a 100-year celebration or something, and but they were very honest about this. So they had a historian coming up, and she explained, she traced everything that was done actually in the 1930s, and I mean, I tell you, it made me cry in the lecture room. I mean, it is just horrible. I mean, Hallervorden was interested in autism and neurodevelopmental diseases, and I mean, he ordered the Gestapo to take the children with mental retardation, bring them to Dachau, close to Munich, murder them, and bring back the brains to him for analysis. I mean, it's just unbelievable. And I think we can never forget these things. And this destroyed neuropathology. I mean, for decades, this was like something that just destroyed the field. And then for decades, nothing happened. And then it was a biochemist in Tübingen, and his name was Konrad Bayreuther. And in the early 80s, I mean, he was really a conventional biochemist. And in the early 80s, he teamed up with an Australian scientist, neuropathologist, Colin Masters. They took the brain of people dying from Alzheimer's disease, and they did the same thing that Manuela Neumann did. They cleaned up the plaques. So they invented, and what they found is that if you treat the brain with concentrated formic acid, which is a very, very corrosive compound, then essentially you dissolve everything except for the plaques. And this was really enabling, because then they could actually centrifuge down the plaques and they could isolate them. But this was in the early 80s, and so there was no mass spectrometry back then. And so they wanted to find out what is in the plaque, and that was very hard to do. And there was a technology that of peptide sequencing that had been invented by Fred Sanger. Fred Sanger is, I think, the only biologist who got 2 Nobel Prizes. One for developing the Sanger sequencing method of DNA, and then afterwards he developed a method for sequencing protein, and then he got a second Nobel Prize. And so that was the method, the Sanger method for very, very hard way of determining protein sequences, and you can also determine short sequences. And so Bayreuther and Colin Masters actually did that, and they found, so they sequenced actually the main component of the plaque protein. And what they found was a protein of 42 amino acids, so a short peptide. And they called it amyloid beta, or A beta. And so So that was, so A beta is the main component of the plaques. And I call it also A beta 42 because it's it has 42 amino acids. And so, okay. Then, so this was a very, very important discovery. And then the question was, okay, where is the gene that encodes for A beta? And now comes a super interesting story. The gene was found, and it turns out the gene is called APP. And APP means amyloid precursor protein. Because A beta is amyloid. So you know what is amyloid? No, it's you can stain it with Congo red. It's a birefringent. You can stain it also with thioflavin. All of this applies to the neuritic plaques. They behave like amyloid, and so when they found the gene, but the gene encodes for a very long protein. So it turns out that if you think, I mean, if you think of the plasma membrane, so the APP gene encodes for a transmembrane protein. It's a type 1 transmembrane protein. So the N-terminus is outside, the C-terminus is inside. This is a type 1 protein. Type 2 proteins are the other way around. And so essentially, if you have the plasma membrane with a lipid bilayer, and here is the continuous plasma membrane, and here you have all the lipid bilayers. And then essentially what you have is the APP molecule looks like this. It has the N-terminus in the extracellular part, it has the C-terminus intracellular part, and but this is like 800 or 780 amino acids. I don't remember, but something in that order of magnitude. But A beta is 42 amino acids. So what was found is that A beta consists of just a small part of this. It's A beta goes from here to here. So this is actually the A beta fragment. So it's derived from APP, but it's a small part of it. And actually, it turns out that there is A beta 40 and A beta 42, and there are actually also many other moieties. So the N-terminus is pretty well defined. The C-terminus is not so well defined. So the N-terminus is always the same. So so very clearly, so you have APP and A beta is a catabolite. It's something that arises from APP. And now this is very interesting because if A beta is the cause of Alzheimer's disease, and this is a big if, but let's assume, and it's still contested, but let's assume for a moment that A beta is the cause of Alzheimer's. And now you would say, okay, I mean, if you can actually, so first of all, can you remove APP? I mean, if you delete APP, will you prevent Alzheimer's disease? Well, first question is, how safe is it to delete APP? Now, or to do anything, any drug that lowers APP, that is a possibility. So how would you go about testing this? Let's say you want to know if if removing a gene creates a problem in an organism. What would you do? Yeah, but what would you in practice? Yeah. Yes, you can make a knockout mouse. Perfect. Very good. So that's exactly what the laboratory of Ulrike Müller and my laboratory and the laboratory of Charles Weisman together did to knock out the gene that encodes APP in the mouse, and to make a mouse that has every gene of the mouse except APP. And the question was, will this mouse have only 3 legs or only 1 eye or whatever? And it turns out that you can take, and we did this 30 years ago, a long time ago, and It turns out, and at that time it made a big splash. It was on the cover of Cell, and the reason was that you can take out APP and not much happens. And and this is super important because it means that if it's not essential, then perhaps you can actually create a therapy around removing or somehow down-regulating APP and A beta. So it's not, because the other possibility was, well, maybe this A beta does Alzheimer if you have too much of it, but it may be a very important protein. And then if you take it out, something terrible might happen. So that was not the case. And that was obviously a milestone. And yeah, and then the gene encoding APP. So the gene encoding APP. What is the chromosomal location of the gene encoding APP? Does anybody know? You can guess. Okay, you just guess. You don't want to guess. I tell you. Chromosome 21. What comes to your mind when you think of chromosome 21? What comes to your mind? What kind of diseases? Yeah. Sorry? Right, trisomy 21. So what is trisomy 21? What happens to these people? That is a thing. You know, trisomy 21 is also called Down syndrome, is developmental disease, and patients with trisomy 21 have a lower IQ, and they are handicapped in many ways. And this is the neurodevelopmental stuff. But then the other thing is that by age 40, pretty much every patient with Down syndrome develops Alzheimer's, and their brain is full of plaques. They have 3 copies of APP. Does it ring a bell? It's the same story as with synuclein. You have 1 copy more, you make 50% more of the gene product, and then over time, the whole thing deposits pathologically in your brain, and it creates trouble. So if you have 3 copies of APP, you get obligatorily with super high penetrance, you get Alzheimer's disease. So that's already a pretty strong hint that A beta may actually be the cause of Alzheimer's disease. Very strong. I mean, when you have this kind of human genetics, it's very hard to argue against them. And so what happens then? I mean, the other thing is, so as I told you, APP is a big protein. A beta is a small fragment of it. So the idea, and so the cut that generates the N-terminus of A beta is very precise. It happens always at the same place, which suggests that there is an enzyme that does exactly that. So there are 3 cuts. One is here, then there is a second cut here in the middle that can happen, and if that happens, this cut here essentially cleaves in the middle of the A beta fragment, so A beta cannot exist. This is a second possibility, so that is actually a beneficial thing. And then there is a third cut, and this is the most interesting one, because this cut here happens in the middle of the membrane. And now the membrane is a highly hydrophobic environment. So you have to realize that enzymes that cut proteins are hydrolases. They use water molecules in order to achieve the cut. Now, water molecules are not supposed to go into the middle of the membrane because there are only lipids there. It's super hydrophobic. So, so then this raised a very crucial question. How do you get this cut? Now, this trick, so let me make it a bit larger. So essentially, so let's say this is A beta. And this is the N terminal. So this would be APP. This would be the N terminal part, the ectodomain of APP. Okay, let's see. This is the plasma membrane. And here this is called the AICD. The AICD is the APP intracellular domain, and what happens here is that when A beta is, this is wrong. There is no cut here. The cut is actually here, and this is the beneficial cut. So the A beta fragment would be from here to here. Yeah. And so when you get this cut, then you generate the AICD. The AICD is delivered into the cytosol. So this is the cytosol, and this is the outside. No, this is the outside. The outside of the cell, the extracellular part. So this is called the soluble APP that is generated. So the soluble APP arises when the APP is cut here. No, then you have the outside domain is liberated. And so what are these three cuts? They are called. So this cut here is called the alpha cut. This is the beta cut. And then this here is called the gamma cut. Now, the alpha cut, as I said, is beneficial. And it turns out that there are many enzymes. It's not very precise. There are many enzymes that can actually cleave APP at the alpha position. And these enzymes are called the ADAMs. And it means a disintegrase and metalloprotease. And so there is ADAM10, is the most important, but there are several others. These are so-called shedases. These are enzymes that essentially cleave proteins at the surface of the cell, depending on the distance from the membrane. So they're very non-specific. They cut pretty much anything as long as it has a certain specified distance from the lipid bilayer. So this is these are the ADAMs. You don't want to inhibit the ADAMs because obviously they do a good job. I mean, they allow you to have actually less A beta, and besides they do a lot of very important things. So you can't touch the ADAMs. I mean, that would be very toxic. Now, the other thing. Beta secretase. Now, so this is called, so the idea is this enzyme, the enzyme that does the beta cleavage, was called the beta secretase, but nobody knew what it was. And but beta secretase is a beautiful target because, because if you can't, if you don't get the beta cleavage, then you don't get APP, you don't get A beta. So clearly inhibiting beta secretase would be a wonderful thing to do. And the third thing is the gamma cut. And this was all of this was happening in the late 80s, and people call this the gamma secretase. The gamma secretase was supposed to be the enzyme that achieves the gamma cut. Now, all of this was very theoretically at the time. So people said, okay, there has to be enzymes that do this job, but nobody knew what these enzymes were. Now, beta secretase. Beta secretase was, so I told you before that the industry is not really capable of finding targets. Beta secretase is one notable exception. It was not discovered in academic lab. It was discovered actually in a company. And now I don't remember the name. I think the company was called Millennium, but I don't hold me on this. I think it was something else. And the guy who discovered this, well, I think he did a fantastic job. But then, interestingly, actually, he moved to the University of Chicago. He didn't stay with the company. So he actually cloned the gene that encodes for the beta secretase. Now, but let me tell you one other thing. There are people with mutations in APP. They have mutations in this region, right after the beta secretase cleavage sites. And these people have enhanced affinity for the beta secretase. That is, these people make much more A beta than normal because of this mutation, and all of them develop Alzheimer's disease by age 35. And so, again, it's not only the Down syndrome people. The people who have the mutation in that enhances the activity of the beta secretase, they also get Alzheimer's disease. The plot thickens. We are getting more and more evidence that A beta is the cause of Alzheimer's. And then there is another story. Then a guy in Canada, Peter Hislop, a human geneticist, found a family of German, the so-called Volga Germans. These were Germans who used to live in Russia, and then after World War II were displaced and migrated to Germany, and they found families with incredibly early Alzheimer's disease. So these people developed Alzheimer's in their 30s, and they had plaques and dementia and everything. It was classical Alzheimer's disease, but very early. I mean, typically Alzheimer's arises when you're in your 70s, 80s. And and it was, again, it was autosomal dominant. And Peter Hislop cloned the gene, and he found actually two families, and they had mutations in two separate genes, and he called them presenilins. Presenilins because this disease was what we call dementia presenilis. So the dementia arose much earlier than normal, and so these are the so he called. And so they are called PS1 and PS2. Now, at that time, nobody knew what PS1 and PS2 did, but what are they? Well, they are 7 transmembrane proteins. So these are proteins that, so the presenilins are proteins that looks like the, now let me see. No, they look like this. So they have 7 transmembrane domains, and and of course people started thinking, okay, maybe the presenilins are the gamma secretase, because nobody knew what the gamma. I mean, it was clear that the gamma cleavage has to be accomplished by some enzyme, but there were 2 big problems with that. First of all, nobody knew what the enzyme was, and second, the cleavage was intramembranal. It was so, and again, I mean, you need water to hydrolyze protein. So how the hell does the water get in there? I mean, it's all lipids. So then it was. Then people started thinking, okay, I mean, maybe the presenilins are actually the gamma secretase, and that would also be a super important target for therapy, because again, I mean, if you block the gamma cleavage, again, you can't make A beta. So it would be, so it's clear. Then you can, if you find an enzyme, then you can, if you know what is the enzyme, then you can devise a small molecule that will enter the catalytic site of the enzyme and block it. No? So that will be the strategy of treating Alzheimer's. So how do you do that? And so is the gamma secretase actually is the presenilin the long sought after gamma secretase? I tell you this. I mean, people went totally ballistic about this. I mean, it was a big fight, and there was a guy in Chicago who he found some evidence that the gamma secretaries would, that presenilin would be the gamma secretaries, but still it was all very unclear and very, very discussed. And then it was a German biochemist, Christian Haas, who actually did a couple of really, really important experiments. And eventually I would give him full credit. I think he is the guy who actually really solved the riddle here. And so Christian, he found, he started looking at homology between presenilin and other genes in the entire, in everything that was sequenced, not only humans. And he found a series of bacterial proteases, which are called the prepilin proteases. And the prepilin proteases are interesting because so they have some homology to presenilins, but very distant. But what the prepilin proteases do is they're intramembranous proteases. So what they do is they're aspartoproteases. So typically a protease uses aspartate residues to immobilize the substrate and to cleave it. And so they're called aspartoproteases. And what the prepilin proteins do is that their active sites look like this. They have two aspartate residues here and here, and these are, and then the substrate comes in, and the substrate is cleaved. The prepilin substrate is cleaved actually because they make a little space, and then that's where the water comes in, and then you have the hydrolytic cleavage of the peptide bond. And so Christian has postulated that this would be the mechanism by which that maybe something similar happens in the presenilins. And it turns out, indeed, that the presenilin have two aspartate residues exactly at the right position, where where you would expect them to be if this was a protease. So how do you do this? Well, what you do is you knock out, first of all, what you can do, you can knock out presenilins, and you can ask, okay, can a beta be processed into, or can APP be processed into a beta through the gamma cleavage in the absence of presenilins? That experiment was done, I think, by Bart de Stroper at the time, and the result is very clear. You knock out the presenilins, there is no cleavage. So that's already pretty good. It does not really prove it, because it could be that the presenilin could be indirect. No, the presenilin could actually act through a completely different mechanism onto a second protein, which in turn metabolizes or cleaves APP. So it does not, I mean, it's a hint. It does not really prove it. The way to really prove it is to do the opposite, to take a cell that cannot process APP, bring in the presenilin, and show that now all of a sudden it processes APP. So what Christian Haas did was to take yeast. So yeast cells, yeast does not have a brain, does not have Alzheimer's, does not have APP. If you express APP in yeast, nothing happens. You just get APP, but you never get A beta. So it does not, so APP clear, so yeast does not contain the machinery that is necessary to do the gamma cleavage. So what Christian Haas did was to take yeast cells, bring in APP. As I said, it's expressed, it's in the membrane, and everything, correct biogenesis goes into the membrane. It doesn't get cleaved, then he co-transfects presenilin and asks whether it gets cleaved. it does not get cleaved. So what do you do then? And well, in the meantime, people have found that actually presenilin exists in the cell, in the human cell, as a complex of 4 proteins. And these proteins, one of the, so there is APP, and then there is a second protein called nicastrin, and then there is a third protein called APH1, and then there is a fourth protein, but I have forgotten the name, and but it's also not so important. I mean, there are 4 proteins, and so what Christian did, and this was really a tour de force, he transfected into yeast all four proteins plus APP, and now you get the cleavage. So you need all 4 proteins, the quartet, it's called the presenilin quartet, and then you actually get the cleavage. And this actually showed that you can reconstitute the entire enzymatic activity with 4 proteins, but you need all 4 of them. And so clearly now this is really the rationale. And so what about the Volga Germans? The Volga Germans have mutations on the presenilin, but most mutation of enzymes, typically what they do is they inactivate. So it's very rare to have a mutation of an enzyme that actually makes it more active. But you would expect the people with the Volga mutation, they end up with more APP, with more A beta. So in some way, so how can this possibly be? I mean, if the presenilin is mutated, you would expect it to lose function, to lose enzymatic function, and then actually you should have less A beta. So how does this work? Well, it turns out that the mutations, they don't actually. What happens? I mean, it's a bit more complicated. There is this cleavage site, the gamma cleavage site happens at position 42, but it can also happen at position 40. And now, depending on where you cleave, if you cleave at here and here at position 42, you get a fragment of 42 amino acids, and this is highly amyloidogenic. This fragment will immediately aggregate and produce plaques, even in vitro. If you make it, you put it into water, or you will immediately aggregate. And the A beta 40 fragment, so the 40 fragment from here to here, is hardly amyloidogenic. It does not aggregate. So I mean, everybody of us makes both A beta 40 and A beta 42. It happens to all of us. But the people with the presenilin mutation, with the pathogenic mutation, the Volga Germans, they make a lot of A beta 42 and very little A beta 40. So the equilibrium between A beta 40 and A beta 42 is completely shifted towards A beta 42. And why is that? Well, it's because, A beta wobbles in the cell membrane. So APP goes a little, it moves a bit up and down, and then when presenilin cuts, it cuts whatever it finds. So if it goes a bit down, then it will cut at 40, and if the molecule goes a bit up, then it will cut at 42. And this mutation, somehow, they modify the geometry of APP such that this favors the cut at A beta 42. So that's more or less how this works. And now, so wonderful. I mean, now we have 2 targets. We have identified them. We have the gamma secretase. We have the beta secretase. Blocking either of them should actually treat Alzheimer's disease. It doesn't. I mean, the problem with the gamma secretase is that, yes, it processes APP, but unfortunately, it processes a lot of other stuff as well. So, first of all, if you knock out gamma secretase, if you try to make a gamma secretase knockout mouse, a presenilin knockout mouse, you can't even get a mouse. I mean, it will die in utero. It's an essential gene. And the reason is that there is a second molecule transmembrane molecule that is very important for development, and this is called Notch. And Notch is also processed by the gamma secretase, and actually in a ligand-dependent manner. So there is, during development, there are ligands to Notch that induce the cleavage, and the cleavage is accomplished by the gamma secretase. And then you have a thing that is called the NICD that results from, what does it mean? What does NICD mean here? Exactly. Very, you're attentive. I like it. And so the NICD is a transcription factor. The NICD, when it is cleaved, goes to the nucleus and activates a program, a developmental program. So this is actually, it's a beautiful system. Now you have an extracellular ligand, it binds to the receptor gets cleaved, the intracellular portion is liberated, it travels to the nucleus, and it activates the transcription of a bunch of genes. So it's a very classical way of how development is controlled. If you block this, development falls completely apart. You can't even get a multicellular organism if you don't have the Notch signaling. So you can't do a knockout mouse because presenil is important for this. And it's even more than that. There is another enzyme that does exactly the same. So it turns out that this intracellular, this intramembranal cleavage is really important. So there is another enzyme, and this relates to secreted proteins. Now, does anybody of you know how what is the biogenesis of proteins that get secreted? I mean, essentially, a protein can do two things. When it is made, it's made by the ribosome, and then it goes into the cytosol, and that's actually normal. So you have transcription, translation, protein is made by the ribosome, stop codon, translation is finished, protein is in the cytosol. How does a protein get secreted? I mean, if it's in the cytosol, it will never get secreted. But how does a cell accomplish the secretion of a protein? Yeah. Yeah, but how does it get into the Golgi in the first place? Very cool. Very nice. Exactly. That's what happens. It's you have the ribosomes attached to the ER. This is exactly the crucial point here. Can anybody elaborate on this? How this works? Exactly. And this is called a signal peptide. And the signal peptide is a short sequence at the amino terminus of secreted proteins that controls exactly this mechanism. And a protein that starts with a secretory signal peptide, what happens is that it gets translated, and then immediately it's a co-translational process. That means it happens at the same time as translation. So the ribosome is attached to the endoplasmic reticulum. All of this is on the cytosolic side. And then when the signal peptide is made, the signal peptide goes into a hole that is actually within the membrane of the endoplasmic reticulum, and then the protein goes into this hole, and it goes into the lumen of the endoplasmic reticulum, and then everything actually goes into the lumen, and this way you can, and then it's inside the lumen, and then everything else is clear. I mean, then it goes to, of course, then from the yard, then it goes into a Golgi, gets glycosylated, then it goes into the endosomal compartment, and eventually it gets secreted. No? So but the crucial moment, the decisional moment, is actually when the ribosome decides to eject the protein into the cytosol, or actually make it pass into the endoplasmic reticulum. And there is a channel there, and it's called the translocon, and it's a complex of proteins. And the translocon has essentially a lumen, and that's the reaction where this whole thing happens. Now, the signal peptide has to be removed. So the signal peptide is only there to get actually the protein into the right place. But then once a protein is started to enter the ER, you don't need the signal peptide more. And there is an enzyme that removes the signal peptide. And this enzyme is called signal peptide peptidase, SPP. Signal peptide peptidase was discovered. So again, for many years, nobody knew what it was. And then there was an Ober assistant here at ETH, just next door at the department, just across the street here. His name was Bruno Martoglio. He cloned the signal peptide peptidase with a paper in Science. It's almost identical to presenilin. And again, it's an intramembranal protease, so it keeps the signal peptides in the middle of the membrane, and it's an aspartate protease. So it's so again, you see actually that all these things are very, very connected, and these are important biological phenomena. Because, I mean, signal peptides, I mean, they are important. I mean, if you can't control where the protein is going in the cell, then you won't even be able to make a cell, not even an organism. And so these are not things that you want to mess around with. And essentially, this is a message that although originally, gamma secretase was thought to be a great target, but it isn't. You can't really touch it. I mean, you get so terrible side effects that it has been tried. I mean, it has gone into patients. I mean, gamma secretase inhibitors have gone into patients, and it makes things worse. The dementia was progressing more rapidly in people with the gamma secretase inhibitors. So complete failure. Yeah, and the beta secretase turned out to be also a very difficult target because it has a pocket. I mean, beta secretase again is a protease, and it has a pocket, but the pocket is very shallow, and this is very important because if you want to inhibit an enzyme, you would want to have, I mean, the catalytic site, the best case is the catalytic site is really something, a deep groove, then you can actually devise a molecule that go inside there and blocks it. If you have a catalytic site that is very shallow, then you can't find a molecule that will really attach to it. And that was a problem, but eventually people were able to make some compounds that would work. It also didn't work very well. And then the third therapy that actually is the most controversial now is antibodies, because then there was the other thing. I mean, maybe you can do the whole thing with antibodies. You can make antibodies against A-beta and then inject them into people, and then they will scoop up all the A-beta, and this should work actually the same thing. And that was again a Zurich story. I mean, a lot of this actually was done in Zurich, and that was the work of Roger Nitsch and Christoph Hock, who founded a company in Schlieren, and it's called Neurimmune, and they made 15 years ago already. So they made antibodies against A-beta, and then they partnered with Biogen, and they made clinical trials, and this was a big story. I mean, the University of Zurich got a lot of millions from this, and Roger Nitsch and Christoph Hock became billionaires, and they don't work anymore, and so it was very successful for them, but it didn't really work in the end. I mean, it went all the way to clinical phase 3, and then there was a minimal signal. It looked like the people who were treated were faring minimally better, but only few people, and then eventually there was a big fight at the Federal Drug Administration. This was maybe 4 or 5 years ago, and then the FDA said they're not going to. I mean, they were giving kind of a conditional license for the drug, but eventually the drug also had a lot of side effects. I mean, people would get hemorrhages in the brain, and so because apparently when you have a lot of A beta, and then the antibody works. I mean, if you measure the amount of A beta, I mean, clearly these antibodies are very effective. I mean, they really clean up the brain, but the patients were not really doing better. And so in the end, Biogen abandoned the program. So you cannot, this antibody was called aducanumab, and you can't buy it in the pharmacy now. And then another company, an American company, Eli Lilly, had produced a different antibody, and their idea was a bit different. They found, I think it was a bit smarter actually. What they found was that the A beta form that deposit into the brain is post-translationally modified, is not just A beta. It's there is a glutamine at the beginning of the sequence that becomes cyclical. It has it is modified. It becomes a thing called pyroglutamate, and pyroglutamate is I mean, as a chemically modified form of glutamine, and then they thought, okay, maybe we should actually make antibodies that bind specifically to pyroglutamilated A beta rather than the wild type A beta, because that is actually what is deposited in the plaque. And so they made this specific antibody, and I should also disclose that I was an advisor to Lilly at the time, and I helped them doing this thing. And they made another, so the antibody was called donanemab, and that worked a little bit better. And eventually it was released, and you can buy donanemab. You can prescribe it to your patients. And the thing is, I have to admit, it works very poorly. I mean, it's the gains are absolutely minimal, and and it costs a hell of a lot of money. I mean, you know, we are talking $25,000 a year or so, and you know, and the clinical benefit is dubious. And that is now, of course, a big fight, because people say, these pharma, they only want to make money. So, you know, they're not entirely wrong, I have to say. And so I mean, just to clarify, I didn't make any money on this. I mean, I just for you to be, so I don't have zero skin in this game. And but yeah, it's difficult. I would say, I mean, my take from this is that from a theoretical viewpoint, donanemab is an important molecule because actually people do fare better minimally, but something happens. And I think that this vindicates the whole hypothesis, the so-called amyloid cascade hypothesis. The amyloid cascade hypothesis says that Alzheimer's disease is caused by an overproduction of A beta, which then will lead to a number of downstream things, accumulation of another protein called tau, and then microglial reaction, and then neuronal loss, synaptic retraction, and eventually dementia. No? So this is called the amyloid cascade. You have the beginning of the cascade, you have A beta, and then you have a bunch of other things. And I would say that the results from the clinic with donanemab showed that the amyloid cascade hypothesis is right. And this is hugely important for theoretical reasons. However, it also says that at least if you have already signs and symptoms of beginning Alzheimer's, removing A beta is not enough. I mean, it may be a part of it, and I think it should be part of any therapy of Alzheimer's disease, but it's not enough. You're not going to block Alzheimer's disease just by blocking A beta. So you have to do more. So but you know, this is true of many diseases. I mean, cancers, most cancers, you cannot cure them with one single chemotherapeutic agent. Also there, you have to do combination therapies. So, so I would say it's nothing earth-shaking. It's something that happens often in pharmacology, but at least we have now some ideas of how to move forward.